discuss about incomplete dominance in snapdragon and grain size in maize. Color flower in snapdragon and grain size, starch grain size in pea plants. Starch grain size in pea plants. And also I will discuss with multiple alleles. Multiple alleles. Best example for multiple allele is ABO blood group, and also different genotypes for blood groups in human beings. Different genotypes or different blood groups in human beings. I will discuss all these concepts in last class. Now I will discuss about codominance. I will discuss about codominance. Codominance. So what is codominance? Codominance is the phenomenon in which the simultaneous expression of the dominant allelic pair of a gene, in heterozygous condition, without blending of tracks, without blending of tracks, is called codominance. Codominance is the phenomenon. Codominance is the phenomenon in which simultaneous expression of dominant allelic pair of gene, in heterozygous condition, without do not blend its trach, is called codominance. Clear? It is called a codominance. See, codominance is the phenomenon in which simultaneous expression of dominant allelic pair of gene, in heterozygous condition, do not blend its tracks, is called codominance. Simultaneous, together, both the dominant alleles of allelic pair of a gene in heterozygous condition, both the dominant alleles can express its character without blending. Both the dominant allelic pair of a gene can express its tracks. It is called codominance. Both the dominant, co means together. Both the dominant allelic pair, can express together, without blending its trach in heterozygous condition, is called codominance. Clear? Both the dominant characters can express equally and produce their characters. Example, AB blood group in man. AB blood group in man. See, the honey IA and the honey IB, the honey IA and honey IB are brought together in hybrid. Both are brought together. Both are brought together in the hybrid. Both are brought together in the hybrid. Brought together in the hybrid. So both the alleles IA and IB, both alleles IA and IB, both alleles IA and IB express their characters. Express their characters. Or express their traits equally. Express both the alleles IA and IB express their traits equally and produce AB blood group and produce AB blood group in man. AB blood group in man. This is the best example. AB blood group in man is best example for codominancy. IA and IB both alleles are equally produce their characters, or traits. Allele IA and allele IB equally produce their traits and produces AB blood group in heterozygous condition. See, these both are dominant. allele IA is also dominant. Allele IB is also dominant. That's why in heterozygous condition, both the alleles IA and IB both are produce equally their characters and produce AB blood group. Clear? So it is codominant, simultaneous expression. Both the alleles IA is also dominant, IB is also dominant. So IA is also dominant, IB is also dominant. These two alleles are brought together in heterozygous condition. Are brought together in hybrid. Both the alleles IA is also expressed, IB is also expressed because both are dominant. That's why they produce the AB blood group. This is called the codominance, and best example is ABO blood group. Next, difference between codominance and incomplete dominance. Difference between codominance and incomplete dominance. Incomplete dominance. Codominance and incomplete dominance. So, codominance, both the alleles, codominance, both the alleles are equally expressed. Both the alleles are equally expressed. Both the alleles are equally expressed. Second one, heterozygotes. Both the alleles are equally expressed. We know, I will discuss in codominance, IA and IB are equally expressed. That's why they produces AB blood group. And heterozygotes form traits. Heterozygotes for traits of heterozygotes shows traits for both alleles. both the alleles. Heterozygotes show traits for both the alleles. So next here, in incomplete dominance, we know what is incomplete dominance. So incomplete dominance in which the one allele is incompletely dominant. Incomplete dominance is one in which one allele is incompletely dominant. One allele is incompletely dominant over recessive. In heterozygous condition or in hybrid, one allele is incompletely dominant over recessive. So the heterozygotes produce intermediate. The heterozygotes produced the intermediate phenotype. I have discussed the incomplete dominance in snapdragon. Pink flower. Color of the flower is pink. Pink is the intermediate phenotype. This is the difference between codominance and incomplete dominance. See, codominance, both the alleles are equally expressed. Best example for codominance is ABO blood group. In heterozygous condition or in hybrid, allele AA and allele IB both are equally expressed and produce AB blood group. And heterozygotes shows traits for both the alleles. So heterozygotes IA and IB traits show traits for both the alleles. The heterozygote shows the characters for both the alleles. Both the alleles shows the character. Both the alleles. IA is also expressed and IB is also expressed. So incomplete dominance. In heterozygous condition, the dominant gene or dominant allele is incompletely dominant over recessive. It is called incomplete. Best example. So color of the flower in slabdroma. Flower color in slabdroma. Heterozygotes produce intermediate phenotype. So intermediate phenotype in incomplete dominance is pink color flower is obtained. It's called pink color of the flower. It's an intermediate. These are the two differences between codominance and incomplete dominance. Next one is next concept is chromosomal theory of inheritance. Next concept, chromosomal theory of inheritance. Chromosomal theory of inheritance. Chromosomal theory of inheritance. Chromosomal theory of inheritance. So the chromosomal theory of inheritance. The chromosomal theory of inheritance was proposed by chromosomal theory of inheritance was proposed by Sutton, SUTTO and SUTTO, Sutton and Boveri, BOVER. Sutton and Boveri proposed the chromosomal theory of inheritance in 1902 to compare Mendelian factors to compare Mendelian factors to compare the Mendelian factors with Mendelian factors with behavior of chromosome, behavior of chromosome, behavior of chromosome, behavior of chromosome. So Sutton and Boveri proposed the chromosomal theory of inheritance in 1902 to compare Mendelian factors with behavior of chromosomes during mitosis and meiosis. During mitosis and meiosis. Mitosis and meiosis. Mitosis and meiosis. Here to compare Mendelian factors with the behavior of chromosomes during mitosis and meiosis is called. So chromosomal theory of inheritance and proposed by Sutton and Boveri. So according to Mendel, law of independent assortment, according to law of independent assortment. This is also called Mendel's second law. Law of independent assortment is also called Mendel's second law. According to law of independent assortment, according to the law of independent assortment, the chromosomes assort independently. The chromosomes assort independently with each other during gamete formation. Independent assortment. So chromosomes first segregate and assort. During meiosis or gamete formation. Clear? According to the law of independent assortment, the chromosomes segregate and assort independently during meiosis or gamete formation. So, at the time of gamete formation or meiosis, the chromosomes are segregated. Segregate is separate. The chromosomes get separated and assort independently during meiosis or gamete formation. This is called law of independent assortment. It's very simple. Chromosomes are get segregate means separate and assort independently during meiosis or gamete formation. This is called law of independent assortment. So Sutton and Boveri so observed independently. Sutton and Boveri, both of them observed independently the Mendelian factors. Observe independently the Mendelian factors and behavior of chromosomes. Clear? So chromosomal theory of inheritance was proposed by Sutton and Boveri in 1902 to compare Mendelian factors with behavior of chromosomes during mitosis and meiosis. So what's the behavior of chromosomes? In mitosis and meiosis. How the chromosomes are behave during mitosis and meiosis. And Mendelian factors. Comparison of Mendelian factors with behavior of chromosomes during mitosis and meiosis by Sutton and Boveri. So according to Mendel's law of independent assortment, the chromosomes segregate and assort independently during meiosis or gamete formation. First the chromosome get separated, segregation means separated, and assort independently during gamete formation. And Sutton and Boveri observed independently. So the Mendelian factors, both of them observed the Mendelian factors and so behavior of the chromosome, and proposed first one the homologous pair of chromosomes, the homologous pair of chromosomes, and Mendelian allelic pair of Mendelian factors segregate. First, so the homologous pair of chromosomes and allelic pair of Mendelian factors are segregate and pass to different gametes during meiosis. So Sutton and Boveri observed independently the Mendelian factors and behavior of chromosome, and proposed first one the homologous pair of chromosomes and allelic pair of Mendelian factors. First segregate means separate. After separation they pass into different gametes. Or they entered into different gametes during meiosis. These chromosomes or alleles means genes or factors, first separate and entered into or passed into different gametes during meiosis. They are entered into the different gametes in the meiosis, during meiosis. So second one, the homologous pair of chromosomes, the homologous pair of chromosomes and allelic pair of Mendelian factor. The homologous pair of chromosomes and allelic pair of Mendelian factor assort independently during gamete formation. These two theories are proposed by Boveri and Sutton. So, the homologous pair of chromosomes and allelic pair of Mendelian factor assort independently during gamete formation. Or meiosis. Yeah, these are the two concepts proposed by Sutton and Boveri by observing independently the Mendelian factors and the behavior of the chromosomes. Clear? So, chromosome theory of inheritance was proposed by Sutton and Boveri. And to compare the Mendelian factors and so behavior of the chromosome, and proposed these two concepts. First one is the homologous pair of chromosomes and allelic pair of Mendelian factors segregate and pass into different units during meiosis. Next thing, the homologous pair of chromosomes and allelic pair of Mendelian factors assert independently during meiosis. These two concepts were proposed by Sutton and Boveri in 1902. Here, this is chromosomal theory of inheritance. Next, the examples for chromosomal theory of inheritance. Example for chromosomal theory of inheritance. Best example for chromosomal theory of inheritance is dihybrid cross. Dihybrid cross in pea plants. By some certain form. Best example for chromosomal theory of inheritance proposed by Sutton and Boveri is dihybrid cross is the best example in pea plants. So, in pea plants, Mendel crossed are considered two pair of contrasting characters. Mendel considered two pair of contrasting characters. Two pair of alleles. Two pair of contrasting characters are two pair of alleles. So these two pair of alleles are present on different chromosomes. These two pair of alleles are two pair of alleles are present on different chromosomes. Different chromosomes which segregate during gamete formation and segregate during gamete formation. See? So here Mendel considered two pair of alleles for two pair of contrasting characters around yellow. Mendel considered around yellow and green wrinkle or yellow round and green wrinkle. So chromosomal theory of inheritance. This example is dihybrid cross in pea plants. So the two pair of alleles are two pair of alleles for different. The two pair of alleles are present on different chromosomes. You know what is dihybrid cross? It's a cross made between two homozygous individuals differing in two pair of contrasting characters. So two pair of alleles. These alleles present on different chromosomes. These alleles are present on different chromosomes and segregate during gamete formation. These chromosomes are segregate during the gamete formation. So Mendel considered yellow, color of the flower is yellow, is dominant over green, and the round, shape of the same, round is dominant over wrinkled. Correct? So then, the homologous chromosomes, these homologous homologous chromosomes. The homologous chromosomes. The homologous chromosomes. The homologous chromosomes. Assemblage. The homologous chromosomes get assemblage. The homologous chromosomes get assemblage in during metaphase. First of meiosis. Metaphase first of meiosis. So the homologous chromosomes get assemblage during metaphase first of meiosis. Metaphase first of meiosis. And these chromosomes segregate or separate. These chromosomes are separate in two possible ways. See, the chromosomes are get assemblage during metaphase first of meiosis and separate in two possible ways. And these aggregated assemblage chromosomes are separate into possible ways. First, the homologous chromosomes are assemblage. Assemblage means aggregate. Clear? The homologous chromosomes are assemblage or aggregate during metaphase first of meiosis. And these chromosomes again separate into possible ways. They are segregate or separate into possible ways. So there are four types. There are four different types of gametes are produced in equal proportion. There are four types of different gametes are produced in. So there are four different types of gametes are produced in equal proportions. Among four, two are parental type, and two are recombination type. Two are recombination type. See, there are four different types of gametes are produced in equal proportion. So, among four types of gametes, two types of parental type and two are recombinant type. See, there are four different types of gametes are produced. Among these four types of gametes, two are parental type and two are recombinant type. In F2 generation, the offspring produced are in the ratio of 9:3:3:1. The offspring are produced in the ratio of 9:3:3:1. And among these fours, 3 and 3, these are the recombinant type. These plants are recombinant type. New combinations. These combinations are not appear in the parental generation. Already I have discussed in the dihybrid cross. This is because the formation of these two recombinant type is because of law of independent assortment. Proposed by Mendel during gamete formation. They first the chromosomes get segregate and assort independently during meiosis. Correct? That's why the chromosomes get assort during meiosis, and it results in the formation of two new combinations. It results in the formation of two new combinations along with parental combinations. See, yellow round, yellow round, yellow round. 9. Green round, green round. This is the recombination type. And yellow wrinkled, yellow wrinkled. It's also recombination type. And green wrinkled, parental type. Green wrinkled is the parental type. 9:3:3:1. So green round and yellow wrinkled are the recombination type. New plants, recombination type, new varieties, the green round and yellow wrinkled are the new varieties are recombination type produced in the F2 generation. It is because due to the chromosomes or get assort independently during meiosis. Yeah, this is the example for chromosomal theory of inheritance proposed by Sutton Boveri. Hybrid cross in pea plants is the best example for chromosomal theory of inheritance. Clear? Next, one is linkage and recombination. Next concept, linkage, linkage and recombination, linkage and recombination, linkage and recombination. Linkage and recombination. Next combination. So linkage and recombination. So genes, genes, genes for different characters, genes for different characters, genes for different characters. Genes for different character are present on same chromosome, are present on different chromosome, different chromosome. See, linkage and recombination. Genes for different characters. So genes can control the different characters. Different genes can control the different characters. So the genes control different characters. These genes may either present on the same chromosome. The genes which control the different characters. The genes can, we know that the genes can control the different characters. The genes control different characters may either present on same chromosome or different chromosome. They are present in same chromosome or different chromosome. On the other hand, so according to Mendel's law of independent assortment, according to the 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 law of independent assortment, when the genes are located, when the genes are located, when the genes are located, when the genes are located on different chromosomes, different chromosomes, assort independently, assort independently. It's very important. See, according to the law of independent assortment, when the genes are located according to the law of independent assortment, when the genes are located on different chromosomes, assort independently. See, the genes are present in different chromosomes. These genes can get assort independently with each other during meiosis. Clear? On the other hand, the genes present on the homologous chromosomes very closely, located very closely, very near on the homologous chromosomes, assort together with each other. So on the other hand, the genes present on chromosomes, very closely, the genes present on chromosomes very closely tend to assort together. See, it is different. See, the genes present on different chromosomes assort independently. But on the other hand, the genes present on the chromosomes very closely. The genes are present very closely, or very tightly, or very near with each other. They tend to inherit together. Clear? So, according to law of independent assortment, so the genes present are located on different chromosomes, assort independently. But on the other hand, so on the genes are present on the chromosomes very closely. Some genes are present very closely, very near, tightly on the chromosomes. These genes are tended to inherit together. So it means the genes present very closely on the chromosomes are very near on the chromosomes. The genes present very near, very close on the chromosomes. They tend to inherit together. Because the genes on the chromosomes present very near, closely. That's why these closely related or closely present or closely related genes can inherit together. Clear? Closely located genes on the chromosome, so inherit together. Clear? So next, so the phenomenon of inheritance of two or more The phenomenon of inheritance of two or more genes. The phenomenon of inheritance of two or more genes. Two or more genes. The phenomenon of inheritance of two or more genes together present in same chromosome. Present in same chromosome. Present in a same chromosome. The phenomenon of inheritance of two or more genes together present in same chromosome from one generation to another generation. From one generation to another generation. Another generation is called linkage. Is called linkage. Is called linkage. So such genes are called linked genes. Such genes are called linked genes. Called linked genes. Such genes are called linked genes. Such genes are called linked genes. See, the phenomenon of inheritance of two or more genes. The phenomenon of inheritance of two or more genes together. The genes, two or more genes are there together, present in the same chromosome. These genes, two or more genes, are present on the same chromosome. From one generation to another generation. It is called linkage, and such genes are called linked genes. Linked genes. And the phenomenon of linkage. The phenomenon of linkage. The phenomenon of linkage was discovered by. The phenomenon of linkage was discovered by T.H. Morgan. Discovered by T.H. Morgan. The phenomenon of linkage was discovered by T.H. Morgan in fruit fly. In the fruit fly. In the fruit fly called Drosophila melanogaster. Drosophila melanogaster. Melanogaster. Drosophila melanogaster. It is also called Cinderella of genetics. Cinderella of genetics. Cinderella of genetics or biological Cinderella. Biological Cinderella. Cinderella of genetics or biological Cinderella. So T.H. Morgan, very first. T.H. Morgan in 1911. So T.H. Morgan, very first in 1911, discovered the linkage in fruit fly. It is called Drosophila melanogaster. It is commonly called Cinderella of genetics or biological Cinderella. Clear? So who discovered linkage? T.H. Morgan in Drosophila melanogaster. What is linkage? The phenomenon of what is linkage? The phenomenon of inheritance. Linkage means the phenomenon of inheritance of two or more genes present on the same chromosome from one generation to another generation is called linkage, and such genes are called linked genes. And very first T.H. Morgan discovered the linkage in Drosophila melanogaster. It is commonly called Cinderella of genetics. Clear? So then these linked genes are get separated. These linked genes are get separated. The linked genes are get separated. The linked genes are get separated. the linked genes get separated. The linked genes get separated by the linked genes or get separated by the linked genes or get separated by so breakage and reciprocal exchange of reciprocal exchange of broken chromosomal segments. Chromosomal segments. See, reunion. The linked genes or get separated by breakage and reciprocal exchange of broken chromosomal segments. So, between homologous chromosomes, between the homologous chromosomes is called crossing over. See, the linked genes get separated. By breakage and reciprocal exchange of broken chromosomal segments between homologous chromosomes. So it's called crossing over. Crossing over leads to genetic recombination. Crossing over leads to genetic recombination. Crossing over. In crossing over, exchange of genetic material takes place. See, the linkage was discovered by T.H. Morgan in Drosophila melanogaster. So, the linked genes get separated by breakage and reciprocal exchange of broken chromosomal segments between homologous chromosomes. It is called crossing over. The crossing over leads to genetic recombination. So, recombination, crossing over, exchange of genetic material takes place. Here, the exchange of genetic material takes place. This is called genetic recombination. Yeah? So, linkage was discovered by T.H. Yeah? So, there are two types of linkage. Linkage. There are two types. One is complete linkage. Second one is incomplete linkage. Complete and incomplete linkage. So, T.H. Morgan proposed the linkage, and again in linkage two types, complete linkage and incomplete linkage, with or examples, complete and incomplete linkage examples taken by Drosophila. Complete and incomplete. So, linkages are explained in the Drosophila. So, next class I will discuss, tomorrow I will discuss about complete and incomplete linkage.